Kia ora, everyone. Thank you very much for giving me the opportunity to come here and talk to you about some of the science that I do. I feel quite humble, actually, that you have actually asked me to come along, because you're doing such great, fantastic work already, looking at your environment, and especially hearing from the younger guys who talked earlier on about their aspirations and that awareness of how we have to protect our environment for the future, and already knowing the problems that might arise and how to actually deal with them. So, yeah, thank you very much for letting me come along. So, my name's Louise Weaver, and I'm a senior scientist at ESR. I work in the groundwater group, and in that role I work as an environmental microbiologist, trying to look at basically the bad guys, so the pathogens that we might find in those environments, but I also have a real interest in who are the good guys. So in these systems, which I'll show you a little bit more about in a wee while, there's a whole complex diversity of organisms that actually help to remove some of these contaminants. So if we understand these more, then we might have a better opportunity to either remove contaminants that are already present there or give an indication of this system is actually stressed. So this is all in groundwater environments that we're talking about today. So groundwater, I'm sure that you know what groundwater is, but just to sort of bring it into context, a lot of people that I talk to in other communities, you're very switched on here, but in some communities they think, well, we've got water everywhere, water's available to everybody, what's the problem? We just have enough of it for everybody. But when you look at like this schematic here, you can see that yes, there is water everywhere, but the amount that we can actually use, which is the fresh water, is only 3%, and so a tiny amount is actually available for us to use. But then of that as well, within it, a lot of it is, do I have to update that? Do you want it updated now? Okay. Yeah, so a lot within that 3% that's available, a lot of it is still tied up in things like ice caps and glaciers, so we can't gain access to it very much either. And within the amount that we can actually access, groundwater is a big predominant area of it. But we know very little about groundwater and the systems that are present there, even though it is part of this whole water cycle that we all rely on. We all need water. Everybody and everything needs water to survive. And it comes as part of this whole cycle of water, and groundwater plays a part in that. But because it's inherently quite difficult to look at and investigate, then there's a big knowledge gap we feel in this groundwater environment. So I just put this on here to give a perspective that we do have quite a lot of water in New Zealand, but different regions have different amounts available to be used. And see that Canterbury region, where I'm from down in Christchurch, we have a lot of water that we use, and a lot of it is allocated to irrigation. Huge irrigation occurs down in the Canterbury region. But even in the Gisborne region, even though you're right down there on the smaller area of the scale, there is still groundwater that you utilise and use, and it is a finite resource. So that's just to bring in perspective again of just remembering again that groundwater is just one part of this whole cycle. So if we impact groundwater, we also end up impacting surface water as well, and oceans, the whole cycle essentially. So as I say, the thing that we find is that groundwater is often out of sight, out of mind, and especially when we go and talk to, which was initially quite surprising to me, talking to some of the councils, they say, well, it's a sterile environment, there's nothing wrong with it. If we pick up an E. coli, then yes, we've got a problem, but other than that, it's fine. But we're trying to raise awareness that no, there is a whole complex ecosystem that is present there that we need to understand more about, and to see how does that system in the groundwater actually remove some of these contaminants, and how can we protect it and make sure that it does this in the future. So I care about water because I'm worried about the contamination that we're actually impacting on it, and as I say, we all need water to survive. So there's lots of different activities that we do that actually impact on our groundwater. And a bit more of a local context, if you So you have your groundwater source and you have your nice well on your land that you might use to top up your water supply for your drinking water, or use it for your irrigation. That's fine and great, and it's been working for all history that you know since you drilled it. It's been working perfectly. But what you might not know, or you might not realise, that somebody else further up or in the same aquifer, they might come along and they might drill a new well, or they might have a consent to increase their irrigation. They might start taking more water. And if we start taking too much water, then numerous effects can occur on each person individually and as the aquifer as a whole, so the whole catchment can be affected. And one of these is that you end up with a dry well, essentially, because your well that was once within the water table is no longer there. So if that occurs, then you have options of either you can't use that well anymore, you have to drill a new one, or you have to drill deeper into that water table. Another thing that can happen as well is that you can get land subsidence, because you're taking more water that is in this aquifer environment, and the land actually will sink because it's no longer being held up by the water that's present in there as well. So all of these issues essentially end up costing money. So that's another consideration that I think is important to think about as well. It's not just protecting the environment as well, but it's also economic, and for the future you want to keep being able to use and access your water, otherwise it's not viable living where you are. Another thing that we've had this happen in Canterbury region, where a new subdivision was put in place where individual septic tanks were in place, and that has an impact on the water sources for other people not on that new subdivision but further downstream, so the aquifer became a bit polluted from increasing septic tanks that were present there. But as I say, the main thing that you can think of as well as the environmental and protecting your place that you live, is the increased costs that will occur if you keep over allocating the water, or the water becomes contaminated. All these things end up costing more money, essentially, because even if you still can gain access to the water, if it suddenly becomes contaminated, you end up having to treat your water, which the cost is on you. So the work that I do, I either go and look at pathogens and see how they travel through aquifer systems and how you can actually get them to be removed, attenuated, taken out of that environment. But I also look at the good guys, so the ecology that is in these groundwater systems, which is very difficult to do, and we tend to pinpoint where we're looking. But I just thought, I don't know if this is going to work, hopefully it will. But this is a little video of a sample that we took from a well down in Southland, which is an irrigation well for on a farm. Oh, no. Go back and hopefully press. No, these are real organisms. So I don't know if you can see, but it looks like it's just, bits of rubbish in the sample, and there is like hair from cattle and things, but there's actually organisms that are moving around in that environment. So you find a huge array of different, from the microscopic up to the things that you can actually see. So these are millimeters across. These are different crustacean species, and there's a worm here who's working really hard, and he'll turn over that piece of wood that's in the sample. But it's just to demonstrate that there's a lot in our water that we don't know about. And when we take our water to drink, we don't think about anything like that. We just expect it to be clean to drink. So that's one of the projects that we're doing at the moment, is who lives beneath our feet, essentially. Are they good or are they bad guys, and how are they actually playing an essential role in that environment and in protecting our water? So we go from, because I'm a microbiologist, I'm interested in these guys, the microscopic organisms, the bacteria, viruses, and such like, but there's a whole ecosystem present there. It's the same as even, think of something like on the African savannah, there's a whole massive array of different animals that all live together and survive in that environment. You have the same thing that occurs in the ground, but we can't see it, so we don't know very much about it. So what we've developed in this project, which is ongoing, so we're still piecing together the pieces, is we tried to develop a way of actually sampling our wells. So we had to make these bags. A couple of my colleagues had to sew all these bags up, which we put in the same material that's in the aquifer into these bags, and essentially means that we can drop them down into a well and we can leave them in place for a long time. The reason that we do that is then we can get bacteria growing in these bags, which are the basis for all of the ecosystems that are present there, so we can get a picture of the primary producers. So those are like on the African savanna. Again, the grasses, that's the equivalent. So those are the ones that other organisms start feeding on. So we put these bags in place and we have to leave them there for quite a long period of time. We usually leave them there for a few months, and then we pick them up and see what microbes are there. What we also do as well is we look at the chemistry that's present there, because there's quite a different community that builds up around even the water chemistry, so how the water changes can affect who's present there. And also the big guys, these are what they call stygofauna, so these are only found in groundwater environments, but a lot of them are like little tiny shrimp. They can be millimetres size, and we don't know very much about these guys at all. NIWA have done some work on them, and we're working on a project together to try and get more information on these. So then I do things like this, where I make nice, pretty graphs, essentially, but from that tells me what type of organism is present there. So each of these different colours represents a different microorganism, a different bacteria in this case that's present. And I can look at these samples at different sites and compare these differences in these pretty patterns, essentially, to get a picture of who is present and how do they change with the water chemistry or with the contaminant that's present there. From this, for the toolbox, what we're developing is a way to say, okay, so your aquifer, is it green? Is it quite happy? It's doing what it should do. It's removing contaminants, and the communities that we know are present there are quite happy and they're doing the functions that they should be doing. Or are they getting stressed? So what happens here is when we find that if you have more contaminants coming into an aquifer, what happens is that the diversity, especially on the microbial level, changes, so you get sudden dominance in a certain type of organism, which can be good, or it can also be detrimental to that environment. So we monitor that, and if you're going towards that point, you might want to start doing something about it, because what you don't want to get to is the point where you're in the red and your communities are no longer protecting your drinking water or your irrigation water. There is also another thing that we think that could be useful as well, as if we understand these organisms that are present in these environments, you've got the potential to actually use these communities to actually recover your aquifer. So in Australia and in America, they've used what they call bioaugmentation. So that gets microbes that they grow up in the lab, and they can put them into a well that's been contaminated and actually regenerates that water source. It's an area of research that's quite early still, and it's a bit, I'd say, hit and miss, but it is still a bit hit and miss whether it actually does anything and work. But there is that opportunity that if you know more about the environment that you're looking at, you've got more chance of protecting it for the future. I think that's it. Yeah. So, if there are any questions? Kia ora.
One question? Kia ora. Kia ora, Louise. I'm not sure, but you'd probably not know our Gisborne case, but we've got an aquifer recharge program, but I'm not so much wanting to raise any issues about that. Over the years, we've tried to reduce the overflow of sewage into our rivers, streams, harbour, and out into Turanganuiakiwa Bay. Note I didn't call it Poverty Bay. I think my question to you is that where does wetlands figure in any of your work? And if so, we're really trying to get our council to look at wetlands as being part of the instead of just a disinfectant and an outfall, that we move on to a wetland. Does that, have you done much work in that area?
I haven't myself. Some colleagues at NIWA have worked on wetland systems. The only part I did a project with them where we looked at wetlands, because that's another area where people you use wetlands to remove nutrients, but there's not been a lot of work in looking at how to actually remove the pathogens. So we started to have a look at whether the pathogens are being removed by wetlands as well, which did look quite promising. But I'd say NIWA probably have done more work in that area of wetlands than we have. So on the wastewater side of things, because I do wastewater research as well as groundwater, we tend to work in the oxidation ponds and the conventional treatment works. We also have a big program called CYBER, so that's the Centre for Integrated Biowaste Research, where we're more looking at if you can remove wastewater discharges into waterways and apply it to the land, what's the safest way of doing that? So, yeah, that probably doesn't answer your question, but I can put you in touch with the right people at NIWA to...
Yes, I think I was just going to lead on and say that if industry wanted to, if we were to offer that water in a commercial way out of the wetland, that's the part I think that I couldn't really get my head around, although there is reuse of water is something we should really look at, and not, instead of just letting it flow out to sea.
I think, yeah, definitely reuse is another good option for the future. Yeah. Thank you.
Louise, I'll have to get back to you with my question. It's just skipped out of my mind. But I wanted to ask Tina a question. What the microplastics, when you're in the mahi you've been doing, I'm just interested to know, have we got microplastics on our beaches?
Our understanding, Aotearoa and in fact much of the South Pacific is relatively a blank slate when it comes to understanding microplastics. The South Pacific gyre was only just tracked last year effectively by Algalita. We've had oceanographers mapping the currents to look at where the likelihood of microplastic contamination is, and then we carried out our first microplastic trawl last year between Ahuriri and Pōneke, but on top of that we also did a national series of beach surveys, and what we found kind of suggests that what the oceanographers predict is on the mark, which is that the West Coast acquires microplastics from the currents, so they can get a lot of international microplastics. The East Coast, the currents tend to not carry it as much, but we still do have contamination. It's just that it comes from us. We can say more likely than not, the contamination that we have is locally driven. And what we found when we did our coastal trawl is that, so if it's carried by the currents, you'll have it all the way along the coast. If it's locally centred, you'll find it at the outfalls of the awa and near the town centres, and that's what we found along the east coastline, was that it's around the town centres and the river mouths that we found a lot of the micro and macro plastics. I'm really interested also in understanding groundwater contamination and aquifer contamination. There's some research that suggests that our stygofauna and the kind of equivalent of plankton, zooplankton, and that does the cleaning of our aquifers and groundwater systems are also ingesting microfibres, plastic fibres, and that's interrupting their systems, and that's a really big concern for us in our region, is whether or not our groundwater is being contaminated, because we have quite a lot of landfill, unregulated landfills that are in the back, which means that all of those things won't be getting filtered, so they could very well be making their way into our ground system. It's a part of the research that I think we should be looking at.